In this video recording, we're going to discuss metabolism and nutrition. Metabolism is the sum of all chemical reactions that occur in the body. Metabolism includes catabolic reactions in which a large molecule will be broken down into smaller constituents. This is usually done by hydrolysis, using water to break a burst apart. When catabolic reactions occur, energy is released that can be used to do work, although some of the energy can be lost as heat. Anabolism is the process by which atoms or molecules can be put together to make larger products, and that process requires energy or consumes energy and is often referred to as a synthesis reaction. It's usually going to involve dehydration synthesis, where water is removed in order to form bonds between other constituents. There are also exchange reactions that aren't listed on this particular slide, but remember that an exchange reaction is going to involve catabolism first, to break apart molecules, and then anabolism to put them together in a different order. There are other chemical reactions that are known as oxidation-reduction reactions, and these are reactions that involve the transfer of electrons, most often electrons from hydrogen atoms. If we look at the chart on the right-hand side, you can see that oxidation and reduction are opposite in terms of the process that occurs, and oxidation and reduction can occur in one of three ways. Oxidation can either be done by removing a hydrogen atom, by losing an electron, or by adding an oxygen atom. Whereas reduction is going to be the opposite. It would either be the addition of hydrogen, the gain of an electron, or the removal of oxygen. So if we look at the first two types of processes that occur with oxidation, because we're looking at the removal of hydrogen or the loss of an electron, that means that bonds are being broken. So those would be catabolic in oxidation, the first two, whereas the addition of oxygen and oxidation would be anabolic, because now we're putting something together. Conversely, with reduction, the addition of hydrogen and the gain of an electron would be anabolic, whereas the removal of oxygen in reduction would be catabolic. Remember, catabolic is going to be breaking bonds. In AMP1, I show a picture in a Kahoot with a cat that has torn apart a roll of paper towels because it's releasing its cat energy. So breaking bonds, releasing energy. There's a mnemonic, oil rig. Oil rig means oxidation is loss of electrons, reduction is gain of electrons, and that mnemonic is true for electrons specifically. Chemical reactions in our body often require the assistance of an enzyme molecule. You'll remember from AMP1 that some enzymes are purely protein. However, most enzymes in our body require the assistance of either a coenzyme or a cofactor. Coenzymes usually come in the form of a part of a vitamin. Cofactors are usually minerals. Coenzymes and cofactors, when they're bonded to protein enzymes, can assist the enzyme either by achieving its proper shape or configuration, because you remember that enzymes have to fit with substrates like a lock and key in order for them to produce a product. However, some coenzymes can also act to assist the enzymes by helping to accept electrons and hydrogen ions from other molecules during oxidation reactions. So there are two specific coenzymes, including nicotinamide adenine dinucleotide, or NAD+, and flavin adenine dinucleotide, which is FAD+. Now, sometimes you'll see FAD+, sometimes you'll see it written as FAD. It depends on whether or not it's has lost an electron or has depends on the number of electrons, ultimately. For our intents and purposes, don't get caught up in the minutiae of that. So NAD+ and FAD+, or FAD, are molecules that act as electron carriers in cellular respiration because they have the ability to accept electrons and hydrogen ions from other molecules during oxidation reactions, and then they're able to transfer those electrons to the electron transport chain, which is the end part of cellular respiration, where they're used to generate energy. So the key points about these coenzymes, NAD+ and FAD, or FAD+, is that they can accept electrons and hydrogen ions during oxidation reactions, becoming reduced to NADH and FADH2 respectively. In terms of their role in cellular respiration, NAD+ is primarily going to be involved in glycolysis, which is the breaking or bursting apart of glucose with the help of water. It's also used in the citric acid cycle, while FAD is primarily used in the citric acid cycle. We're going to be looking at this cycle in a short while. So the reduced forms of these molecules is NADH and FADH2. Those are the reduced forms of the coenzymes NAD+ and FAD+, or FAD. Because we're working our way towards cellular respiration, we're going to look at the metabolism of carbohydrates next. And you'll recall from AMP1 that carbohydrates come in different forms. We have monosaccharides, which are single sugars. Glucose is a great example of this, and it's a simple, immediate form of energy. We have disaccharides, which can be broken apart into monosaccharides. And then we have polysaccharides, which is how we normally store excess glucose. You'll recall that glycogen is how we store glucose molecules that are bonded together, and glycogen is stored mostly in our liver and muscles because glycogen can be broken down into glucose molecules, but it is a much more complex stored form of energy than just glucose. So the energy in glucose is found in the bonds, just like it would be for other hydrocarbons like gasoline and other fuels like ethanol. Releasing the energy that's stored in glucose has to be done in a stepwise process. And we remember that carbohydrate digestion begins in the mouth with the assistance of salivary amylase, and that acts on the starches that we ingest, and it ends up with monosaccharides that are absorbed across the simple columnar epithelium that has a microvilli brush border on its surface to increase absorption of nutrients within the small intestines. Now, if all the bonds were broken and the energy was released at once, that would be a bad thing in our bodies. It would be like an explosion. We don't need that. So that's why when we release the energy by breaking bonds in a step-by-step process, it allows us to have a much more organized chemical process to produce energy. You'll recall from AMP1 that the production of adenosine triphosphate, which most people refer to as ATP, is mostly performed in the mitochondria. You'll remember that the mitochondria is an organelle that has two membranes. It's got an outer membrane and an inner membrane. The inner membrane in the mitochondria is where we have a lot of different enzymes embedded, just like we have proteins embedded in plasma membranes on the surface of the cell. In the mitochondria, we also have enzymes embedded on that inner membrane to help these chemical reactions occur. So with the process of cellular respiration, producing a lot of adenosine triphosphate with the use of glucose and oxygen, understand that there are three main steps of this process that we call cellular respiration. The first is glycolysis. Glycolysis, we're taking a molecule of glucose and breaking it down. Then we're going to go through a process known as the citric acid cycle, which is also known as the Krebs cycle. And then the third main step of cellular respiration is the electron transport chain, with that chain being the final stage where most of the adenosine triphosphate is going to be produced by oxidative phosphorylation. Okay, so we know what oxidation is. Phosphorylation basically means we're going to stick a phosphate group onto a molecule. We've seen adenosine diphosphate in the past, and adenosine diphosphate is a molecule with 2 phosphate groups. Adenosine triphosphate is a molecule with 3 phosphate groups, and the third bond for that third phosphate group is a very high energy bond. This is why we use adenosine triphosphate as our form of cellular chemical energy. The first step of cellular respiration is glycolysis. Glycolysis literally means breaking or bursting apart of a molecule of glucose. This is an aerobic process that occurs in the cytoplasm of the cell, and it's actually one of the oldest metabolic pathways. Every living thing does it. And we have what we call substrate level phosphorylation, and we also see that the NAD+ and FAD+ carriers are involved. So if we take a look at this chart at the top, you can see that one glucose molecule with 6 carbons is going to come into the cytoplasm of the cell, and at this point we actually have to use 2 molecules of adenosine triphosphate, where those molecules of adenosine triphosphate will lose that third phosphate molecule, and the glucose becomes phosphorylated with 2 phosphate groups. You can see that right here. Here's one phosphate from the ATP over here, and then there's another phosphate group here. So we've got this molecule that is now phosphorylated, and that becomes glucose 6-phosphate. Now, after that has been phosphorylated, this molecule will split into 2 3-carbon molecules, and electrons from each one will be transferred to NAD+, reducing it to NADH, and that happens with each one of these 3-carbon molecules. And then with the release of the electrons, that drives a free phosphate to bind with those 3-carbon molecules. So now we've got 2 phosphates on each 3-carbon molecule. And now those phosphate molecules can be used to phosphorylate adenosine diphosphate. So we've got 4 total, 1, 2, 3, and 4. So we can phosphorylate 4 adenosine diphosphate molecules and produce 4 molecules of adenosine triphosphate. And once we've lost those phosphate groups, we end up with 2 molecules of pyruvate or pyruvic acid. Now, we used 2 molecules of ATP to drive this process, but we produced 4 molecules of ATP at the end of glycolysis. So after all is said and done, our net gain is 2 molecules of ATP and 2 molecules of pyruvate or pyruvic acid. And the pyruvate is going to be key in the next step. Before we move on, I just wanted to point out that we require enzymes for these reactions to take place. So I'm just circling all of these different enzymes that are required in these chemical reactions, that without these enzymes, these reactions would not be occurring. So we've got all these different enzymes that are required just for glycolysis, okay? I simplified it in the previous slide, and I did not want to lose sight of the fact that without these enzymes, this reaction would not be occurring, okay? Cellular respiration requires oxygen. If there's no oxygen present, anaerobic respiration will continue. It provides a fast resource for adenosine triphosphate, but it doesn't make very much. So it's not efficient for long term. It's only effective for short periods of time. Pyruvate can be converted into lactic acid. Lactic acid is converted back to pyruvate or glucose in the liver within seconds. If oxygen is present, however, pyruvate can be transported into the mitochondria where it will be converted into acetyl coenzyme A, which then enters the citric acid cycle. Each pyruvate molecule that's produced has to be converted into acetyl coenzyme A. In order for that to occur, the pyruvate has to be decarboxylated. It has to lose carbon, and that process of decarboxylation doesn't just remove carbon, it actually removes 2 atoms of oxygen. So we're removing carbon dioxide from pyruvate. That gives us a 2 carbon fragment intermediate molecule which will be joined with coenzyme A, and when they're joined together, a hydrogen atom will be given off in the process, and that hydrogen atom will join with NAD+ to be reduced to NADH. NADH will actually become part of the electron transport chain, which is the big kicker in terms of cellular respiration for producing adenosine triphosphate. Now we have acetyl coenzyme A that can enter into the Krebs cycle or citric acid cycle. And so we see acetyl coenzyme A here with oxaloacetate added to it with the help of the enzyme citrate synthase will form citrate. Citrate with a aconitase will be converted into isocitrate. Isocitrate will also undergo decarboxylation, and it will lose an entire molecule of carbon dioxide in the process. And then that substrate molecule, isocitrate, that's been decarboxylated and has lost its carbon dioxide, with the help of isocitrate dehydrogenase, will lose hydrogen, and that will join NAD+ to form NADH. So NAD+ gets reduced, and now we've got more NADH being made. Now we have the substrate alpha-ketoglutarate, which will also undergo decarboxylation, and we're getting rid of carbon dioxide. And that substrate molecule will interact with alpha-ketoglutarate dehydrogenase. Now we're getting rid of more hydrogen to form more NADH. And now we have this substrate molecule known as succinyl coenzyme A, with which the help of succinyl-CoA synthetase will perform what's known as substrate level phosphorylation to phosphorylate, or that means we're putting on a phosphate group onto guanine diphosphate to make guanine triphosphate. So substrate level phosphorylation basically means we're directly transferring a phosphate group on from a substrate molecule, in this case the succinyl coenzyme A is the substrate molecule, and we're transferring phosphate onto guanine diphosphate to turn it into guanine triphosphate with the help of succinyl coenzyme A synthetase. Now we end up with the substrate molecule succinate, which is going to interact with succinate dehydrogenase, and now we are reducing FAD to form FADH2, and now we have the substrate molecule fumarate, which with water and the help of the fumarase enzyme will produce malate. Malate with the help of malate dehydrogenase, see how we're getting rid of these hydrogen atoms, is going to form NADH, and that takes us back to oxaloacetate with the combination of acetylcholinease is going to be recycled with the help of citrate synthase to take us back through the cycle again. So as long as we have pyruvate molecules that can be transformed into acetylcoenzyme A, we can keep going through this citric acid cycle, and each time we go through the cycle, you'll notice that the products are a molecule of adenosine triphosphate. Why do we say that? Well, because we can easily convert guanine triphosphate into adenosine triphosphate by substrate level phosphorylation to take the phosphate group off of guanine triphosphate and move it onto adenosine triphosphate. Excuse me, move it onto adenosine diphosphate to turn it into adenosine triphosphate. We also get 3 molecules of NADH and 1 molecule of FADH2 and 2 carbon dioxides. Remember, when we go through cellular respiration, we produce carbon dioxide. Where is the oxygen? Because remember, we need oxygen for cellular respiration. Well, that's going to show up in the next part where we go through the electron transport chain and oxidative phosphorylation, which is different than substrate level phosphorylation. So now we're at the electron transport chain, and the electron transport chain is going to use electrons from the previous reactions to create a hydrogen gradient. Now, the electron transport chain consists of a series of proteins, and there's a bunch of different proteins with different names that are embedded in the cristae of the mitochondria. And remember that inside the innermost area of the mitochondria, we call that the mitochondrial matrix. And then there's a space between the cristae and the outer membrane of the mitochondria, because all mitochondria have 2 membranes. They have an outer membrane and an inner membrane with folds called the cristae. So what happens is the NADH that we produced in the citric acid cycle, remember that that molecule was created by reduction. We reduced NAD by adding hydrogen. The converse of that reaction is oxidation. Okay, so we had reduction to create the NADH. Now we're going to go through oxidation when we go through oxidative phosphorylation. And so the proteins that are embedded in the cristae are going to have enzymes that are going to oxidize the NADH, and they're breaking it apart essentially. And what's going to happen is it's going to turn into NAD that is positively charged. It's positively charged because it's lost one electron. So if it loses negativity, it becomes positive. We also have a free hydrogen ion that is positively charged because it's lost its electron. And if we've lost 2 electrons in this process, then we're going to have 2 free negative electrons. Okay, so that's an E. So we get 2 free negative electrons in the process of oxidation. So if we look at this picture, you see these little yellow stars that are associated with the proteins. Those represent the free electrons, and we can see them down here as well. So we've got positively charged hydrogen ions, and those are sometimes referred to as protons. Protons are positively charged ions. They're referred to as protons because that's all that's there. Remember, a normal hydrogen atom is simply 1 proton and 1 electron. So once that electron goes, it's a proton. If a molecule tends to give up a hydrogen ion, that's usually referred to as a proton donor. Acids are proton donors. So taking a look at this, we can see that within the matrix of the mitochondria, we've got 2 free hydrogen ions, and then we've got 2 free electrons, right? And once the electrons are separated from the NADH, the electrons stay in the matrix. However, the hydrogen ions, or protons, are going to go through the proteins that are performing this oxidative process. And so they're going to go across the membrane, and they're going to be collecting in the intermembrane space between the cristae and the outer portion of the mitochondrion. And they're creating, at this point, an electrochemical gradient that will move through a special protein enzyme, and this enzyme is referred to as ATP synthase. Remember, something that synthesizes is going to make something, so it's basically making adenosine triphosphate. And it does that because once the hydrogen ions flow through this protein enzyme, the motion of those is going to drive the addition of an inorganic phosphate group onto an adenosine diphosphate molecule, creating adenosine triphosphate. Where does the oxygen come in? Well, this is where it comes in. Remember, in the matrix, when we oxidized the NADH into positively charged NAD, a hydrogen proton and 2 electrons, well, as we breathe oxygen, it's not a single atom of oxygen. It actually exists as a molecule of oxygen, which is O2. So half of an oxygen molecule is going to accept these free electrons along with 2 of the protons from hydrogen to create water. And that is how the electron transport chain and oxidative phosphorylation work. So remember, oxidative phosphorylation is accomplished because we have this electron transport chain and oxygen creating an electrochemical proton-motivated gradient that powers the enzyme ATP synthase to produce adenosine triphosphate from adenosine diphosphate and inorganic phosphate. And this is actually a much more efficient way to produce a lot more ATP than by glycolysis. Now, you see the term chemiosmosis at the bottom of this slide, and that's basically the motion of the protons. Okay, remember the hydrogen ions are sometimes referred to as protons. So chemiosmosis is the movement of chemicals, chemiosmosis, the movement of chemicals in this instance are protons, that's going to cause the enzyme to become activated to do its job. So the chemiosmosis, again, it's the motion of protons activating that ATP synthase. Gluconeogenesis is the process of creating new sugar. And glucose synthesis normally occurs in the liver by the liver utilizing other molecules and taking those pieces from those molecules to make glucose. So it normally uses pyruvate, lactate, and glycerol to make new glucose molecules. This is going to occur when glucose levels are low. If somebody is fasting or starving or just eating a low-carb diet and sugar levels fall, the pancreas will produce and release glucagon. Remember, the beta cells in the islets of Langerhans are going to produce and release insulin, but the alpha cells in the islets of Langerhans in the pancreas are going to produce and secrete glucagon when blood sugar levels are low. Now, that does different things. One of the things it will do is it will stimulate the liver. The glucagon will stimulate the liver to break down glycogen. That's glycogenolysis. And it will also stimulate the liver to produce more sugar, and that's gluconeogenesis. Triglycerides in the body are either ingested as food, or they can actually be made by adipocytes or hepatocytes by precursor carbohydrate molecules. And we know that triglycerides can be broken down into monoglycerides in the small intestines. The process starts with a tiny little bit of lingual lipase in the mouth, with also a little bit of gastric lipase from the stomach. The liver contributes to this process by secreting biliary salts, as does the gallbladder. The gallbladder and the liver are going to contribute biliary salts through bile, which helps to break down and emulsify those triglycerides. We also have the pancreas with its pancreatic lipase that's going to contribute to this picture to break down the triglycerides so that they can be absorbed, because we can absorb monoglycerides, which are the parts of triglycerides. We can break them down into smaller parts, and those can be transported across the intestinal membrane, so into the simple columnar epithelium. And once it's in there, remember that the intestinal cells will produce molecules known as chylomicrons. Chylomicrons are a combination of triglycerides, phospholipids, cholesterol, and protein that are formed in those cells. And from there, they'll be transported into lacteals. Lacteals will transport those chylomicrons to the liver, where they may be used for other synthesis reactions, or they might be stored as fat. Let's talk a little bit more about chylomicrons. Those are large lipoprotein particles that help transport dietary fat from the intestines to the rest of the body. And that means that chylomicrons are the initial step in the process of fat transport, which can later be processed into what we call low-density lipoproteins and then potentially removed by high-density lipoproteins. So low-density lipoproteins are where there's a molecule that has a fat to protein ratio that's higher fat, less protein. It's considered the bad cholesterol that's coming from the liver to the arteries, and it could potentially build up in the arteries. Now, there is some controversy about that, but we're going to stick with it for now. High-density lipoproteins are considered the good cholesterol that help transport cholesterol from the peripheral tissues, from the bloodstream back to the liver. And again, scientists are really starting to question, the real effects of the LDL and the effects of dietary cholesterol as being bad. More scientific articles are showing that cholesterol is very, very necessary in dietary form. Even though we can produce it in our bodies, it's actually becoming more and more prevalent that we need to eat cholesterol. When it comes to breaking down these lipids, triglycerides can be hydrolyzed using water to lyse or break burst apart into fatty acids and glycerol. Each triglyceride molecule consists of 3 fatty acid tails, which are basically 16 carbon long chains that have hydrogen atoms attached to their periphery. And the triglycerides can be converted to acetyl coenzyme A, which is really useful when glucose levels are low. We can actually use triglycerides to make acetyl coenzyme A so that we can continue with the citric acid cycle. If we look at this particular chart, you can see proteins on the far left are broken down into amino acids. Amino acids can be used to rebuild tissue proteins, and the nitrogen portion of the amino acids can be used to make urea. However, the urea can also contribute to the citric acid cycle. There are ways that that can happen. And then we see that carbohydrates are going to contribute to the citric acid cycle. We already went over that. And as I mentioned just in this slide, if you look at the fats and lipids on the far right, you see that fatty acids can be converted into acetyl coenzyme A. They can also be reconverted into fats and lipids. So here's the whole chain that occurs with lipolysis. If we break down the triglycerides here on the right side, we break them down into fatty acids. That becomes something that can be converted into acetyl coenzyme A. And then we also get some amino acids with that, like phenylalanine, tryptophan, tyrosine, lysine, leucine. And again, it's the acetyl coenzyme A that can go through that citric acid or Krebs cycle. Ketogenesis is the creation of molecules known as ketone bodies, which occur when fatty acids from adipose tissue are broken down and oxidized as fuel to produce acetyl coenzyme A. And when glucose levels are low, oxaloacetate is going to be used to produce glucose instead of merging with acetyl coenzyme A. And that leads to an overaccumulation of acetyl coenzyme A, and those molecules will start to be synthesized together, merged together to form what we refer to as ketone bodies. And this occurs when a person is experiencing prolonged starvation or uncontrolled diabetes. It happens in the mitochondria of the cells of the liver, And then those ketone bodies are going to act as an alternative source of fuel where they'll be exported by way of the bloodstream to the brain, which can use them as fuel. The brain also uses glucose normally as fuel, but if there's no glucose, we'll use ketone bodies. And it's important to understand that the liver is a really important organ in terms of bile production for emulsification of fats, as well as for the metabolism of carbohydrates, lipids, and proteins. Organs that are normally dependent on glucose can use ketones as an alternative source of energy. Because ketones can be broken back down into acetyl coenzyme A if there's not too many of those molecules. And if a person has a huge buildup of ketone bodies, the excess ketone bodies can be broken down into carbon dioxide and acetone, which will be exhaled, giving a person a very sweet alcohol type of odor to his or her breath. And understand that if a person has excessive amounts of carbon dioxide, that can lead to a condition known as ketoacidosis in diabetics because carbon dioxide is acidic. As far as protein metabolism goes, proteolysis, which is the breakdown of protein, occurs initially with the enzyme pepsin and hydrochloric acid in the stomach. Remember, pepsinogen is what is initially produced and that is cleaved by the hydrochloric acid into the active enzyme pepsin. We also have secretin and cholecystokinin in the small intestines that will help break down proteins, and trypsin and chymotrypsin from the pancreas can help break down proteins. When proteins are broken down, remember, the building blocks of proteins are amino acids. So the amino acid constituents of the proteins are going to be transported for use in making new proteins or for producing fats or acetyl coenzyme A. One of the byproducts of breaking protein down is that there are nitrogen groups which are in amino acids, and as amino acids are broken down, which is going to occur mostly in the liver and a little bit in the kidneys, the amino acids will contribute to the production of ammonium. And this is due to what we call oxidative deamination, where the nitrogen is removed from the amino acid. And it also involves transamination, which is the exchange of either an amine group or an ammonium molecule with a keto group, creating a molecule for the Krebs cycle and ammonium for the urea cycle. So when we mix ammonium with carbon dioxide, that creates urea and water. Digestion and absorption is going to last up to approximately 4 hours after eating. And that's the phase of digestion that's referred to as the absorptive state. So during this state, insulin is the main hormone that is directing activities for the absorptive state. And this includes the release of insulin and the conversion of glucose. There's more conversion in cells because there's going to be a higher concentration gradient of glucose in the bloodstream that helps keep glucose moving into the cells. But we've got to have the insulin receptors on the surfaces of cells to move glucose into them. So if energy is exerted, then newly ingested molecules will be used immediately. If no energy is exerted, excess glucose is going to be stored as either glycogen. After our glycogen stores are filled, then it starts getting converted to fat or triglycerides. The post-absorptive state occurs after digestion, absorption, and storage, and the body initially is going to rely on glucogen for energy and then glycogen for energy. Glucagon is a hormone from the alpha cells of the islets of Langerhans in the pancreas that is going to cause glycogenolysis. It's the breakdown of glycogen into glucose, which then now we've got a very distinct pathway to get glucose. We just break down glycogen. Glycogen molecules are just chains of glucose molecules bound together. And glucagon also causes gluconeogenesis, which is making glucose from amino acids and glycerol. Starvation occurs when a person is deprived of nutrients for an extended time. The body enters what's called a survival mode. The priority for nutrition is the brain. The second priority for the body is in conserving amino acids for proteins. And ketones are going to be used for energy because the person's not getting enough carbohydrates or fats or proteins. And fatty acids, triglycerides, are the second form of energy that the body will use. So initially, ketones are going to be produced and used. Secondarily, fatty acids and triglycerides will be used. And then proteins are broken down as a last resort. And you can see in this picture how this person has lost so much muscle mass and bone mass, bone density, because there are proteins everywhere in the body. Remember, collagen is the most abundant protein in the human body. We have collagen in bones. We have collagen all over the body and connective tissue. The body has to regulate temperature very specifically, because if a person gets too hot or too cold, then that's going to disrupt homeostasis. So when body temperature falls, temperature receptors in the hypothalamus are going to stimulate heat-producing mechanisms. And that means that the superficial arteries and arterioles and capillaries are going to constrict to reduce heat loss to the air, and it's going to get blood flowing to our internal organs. A person will also be stimulated to start shivering because the contraction of skeletal muscle can produce heat. And the thyroid gland will also be stimulated to increase the production and release of T3 and T4 because those are metabolic hormones. And so we get these chemical reactions that can produce heat. Negative feedback mechanisms, the hypothalamus is involved, and that can help to raise body temperature if body temperature falls. Conversely, if body temperature gets too high, again, thermoreceptors in the hypothalamus are going to start mechanisms that help cool us off. For instance, we're going to have more blood flowing to the surface of the skin, so our heat can radiate from the surface of the skin. Sweat glands will be stimulated to produce more sweat so it can evaporate off the surface of the skin, and the thyroid gland will decrease its metabolic heat-producing hormones so that our body can cool off a little bit. Again, there are limitations of matter, so we can only cool off to a certain degree. We can only raise our body to a certain degree, and then we're beyond the limitations of matter where the body can start to shut down. So thermoregulation is extremely important. In terms of how heat can be exchanged, conduction is how heat can be transferred by two objects that are in direct contact with one another. This is only about how 3% of our body heat is lost. Convection is the movement of air surrounding the skin. So the warmed air from our body will rise away, and then anything that blows by it can remove that heat and move it away. About 15% of our body heat is lost that way. We can actually radiate and we can lose heat by radiating waves of heat from our body. And radiation is how we lose 60% of our body heat. If we are sweating, then the evaporation of the liquid from our skin will help us lose about 20% of body heat. When it comes to rates of metabolism, as adults, the energy that we consume should be equal to the energy that we expend. If you've ever heard of the term basal metabolic rate, that stands for the daily amount of energy that a person expends at rest. And it's the energy that we need for daily activities. But the more active a person is, the more muscle mass that individual has, the higher basal metabolic rate that individual will have as well. So increasing muscle mass will also increase basal metabolic rate. The nutritional calorie is defined as the amount of heat it takes to raise a kilogram of water by 1 degree Celsius. On average, a person needs 1,500 to 2,000 calories per day to sustain or carry out daily activities. Usually men have a higher muscle mass, so they can consume more calories per day. The breakdown of carbohydrates is what requires the least amount of energy in terms of our chemical reactions. Processing proteins, on the other hand, demands the most amount of energy. And that looks like some really tasty sea bass on that plate. In terms of obesity, obesity is defined by the body mass index, which is a measure of a person's weight in kilograms divided by his height in meters squared. A person is considered to be overweight if that individual has a BMI of 25 to 29.9 kilograms per meter squared, and obesity is considered to be a BMI greater than 30 kilograms per meter squared. This can occur from a number of factors: overeating, poor diet, person who's sedentary, not getting enough sleep. Genetic factors can contribute to this. Diseases can cause a person to become obese. Drugs can cause obesity. Obesity can lead to serious medical conditions like coronary heart disease, type 2 diabetes, endometrial breast or colon cancer, hypertension, dyslipidemia, which is a condition of high cholesterol or elevated triglycerides, stroke, liver disease, gallbladder disease, sleep apnea, respiratory diseases, osteoarthritis, and infertility are all possible complications of obesity. According to most medical standards, obesity is generally defined as having a body fat percentage exceeding 25% for men and 30% for women, meaning someone is considered obese if his body percentage is above threshold. Vitamins are organic compounds found in foods, and they're a necessary part of the chemical reactions that occur in our body. Most vitamins act as coenzymes that are needed to help the body utilize essential nutrients. Remember, coenzymes help enzymes do their job. B vitamins play the largest role of any vitamins in metabolism. However, we need all kinds of vitamins. We need C for collagen. Fat-soluble vitamins include A, D, E, and K. Those are absorbed in the intestinal tract when they're absorbed in conjunction with lipids in chylomicrons. That means if you wanted to have more vitamin A because we need it to be able to see well at night, remember it's involved in the regeneration of retinol in our eyes, it's best to eat fat-soluble vitamins with some form of fat. And minerals in our food are inorganic compounds that work with other nutrients, again, to ensure the body functions properly. Remember, vitamins are coenzymes for enzymes. Minerals are cofactors for enzymes, so they help the enzymes in our body do their job. The most common minerals in the body are calcium and phosphorus, both of which are stored in the skeleton as hydroxyapatite, so they're necessary for bones to be hard. Collagen keeps bones strong, minerals make it hard, and we need water-soluble vitamins, like I mentioned, B vitamins. If a person is taking a particular B vitamin, it's best to use a B complex because B vitamins can compete with one another. So I always tell people, look for a good B complex instead of just taking B12 or B6. Water-soluble vitamins like C, vitamin C, are very important for our body as well. And we get vitamin C from citrus fruits like oranges and grapefruits. And C is important in producing collagen. Collagen, is that a C? C stands for collagen. Okay, let's try to make a C. Again, these nutrients are really important for our body to be healthy, so it's important to eat the right things. In terms of minerals, we mentioned calcium and phosphorus, but potassium, magnesium, zinc, copper, selenium, these are all important minerals that we need for our bodies to function properly. So it's really important to have a healthy diet and to get the nutrition that's needed for these chemical reactions to occur properly. That's it for metabolism and nutrition.